Hello, good afternoon, ma'am. Am I audible, ma'am?
Yeah, good afternoon.
Sorry, ma'am. We were waiting for you, but we were unable to locate you. Sorry for that.
No problem.
Okay. I, Dr. Ruchika Joshi from Physics Department, welcomes you all in this fruitful session of Second Day International Conference on Integrative Chemical Science for Health and Environment, ICHE 2023. So I will welcome you all, and this session chairperson is Professor Gitanjali. Welcome, ma'am, in this session.
Thank you so much.
I am delighted to introduce Professor Gitanjali at the Department of Chemistry, Kirodimal College, Delhi University. Her research interests are in the interference of chemistry, biology, biomaterial. She has done her PhD in organic chemistry. During her 16 years of teaching experience at Delhi University, she has received numerous prestigious awards. Professor Gitanjali is actively involved in scientific publications and editorial activities. She serves as an editor for esteemed high impact journals. Her impressive publication record, editorial role in reputable journals, and leadership in independent research projects underline her standing as a distinguished feature in the scientific community. Her remarkable research portfolio is understood by the publication of more than 46 research papers, authored 15 books, 19 book chapters, 10 modules for EPG Pataksla, completed 2 research projects, and contributed in more than 20 synopses and seminars. Her dedication and expertise promise to significantly impact the academic and research landscape, contributing to a better and healthier world for all. Thank you, ma'am, for giving us your precious time to be a part of our conference. So, ma'am, we will start our presentations by our candidates.
Yeah, Ruchika, thank you so much.
So, ma'am,
you can start.
Thank you, ma'am. Thank you. With your due permission, we will call upon our first candidate. That is, I feel Meenakshi, ma'am is there.
Monica bhi hai.
Monica, she's in, yeah. So, Monica, ma'am, you can start your presentation. Are you there, Monica, ma'am?
Good. Yes. Very good evening to everyone. Hello.
Yeah. You are
Hello.
audible. Hello.
Okay. Can I start?
Yeah. Sure. You can.
Okay. Thank you. Okay. My topic is for presentation is dissipation of ethyl fluorine herbicides in differentially texture soil and conformation of herbicide residues by using GCMS technique. I'm Monica from Department of Chemistry, pursuing my PhD under the guidance of Dr. Anil Duhan in CCSHU University, Hisar. My research topic is ethyl fluorine is a herbicide and very persistent herbicide and widely used in cotton crop like potato crop. As we all know, both cotton crop and potato crop, India highest. India is uh, most leader in terms of area. India stands first in area wise in production of both crops, and we all know that various herbs. Sorry, wait, losses in production of potato and cotton crops are due to infestation of like chenopodium weeds, trianthema, trivinali, cypress. So it is as we know. that India is highest populated country, so land is decreasing, so use of herbicide is must to increase the production. So in the due time, we know that these herbicides are persistent. Ethyl fluorine herbicide is widely used and it is highly persistent in soil. I have taken two types of soil. First is clay loam and another is sandy loam soil. And taken for three replications with two doses. First is standard dose and other is double dose. Then collected samples from the applications with in duration 0 hour, 0 day, first day, third day, seventh day, 10th day, 15th day, 30th day, 45 day, and 90th day after treatment. So then collected soil samples were extracted by using MSPD technique. MSPD technique is metric solid phase dispersion technique. In this, the samples I have collected is taken in petri dish. Then I have added silica gel and charcoal. Then mixed properly, and then a column is pegged, which is sandwiched between uh, layers of anhydrous sodium sulfate by gentle tapping. Then I evaluated the samples with 150 ml acetonitrile. Then extract was evaporated by using rotatory vacuum evaporator up to 5 ml. Then analyzed on GCMS. And before I have done the experiment, I have also done the recovery experiment in which I have seen that all the fortification level I have taken. One is zero point 0.1 microgram per gram, another is 0.01 microgram per gram, and last one is 0.005 microgram per gram. In both the soil, clay loam as well as sandy loam, recoveries are above the 80%. So uh, above 80% recoveries are acceptable. So this method is good for the extraction for the ethyl fluorine herbicide. Then I have observed that recoveries are higher in sandy loam soil as compared to clay loam soil because of the light texture of sandy loam soil, as clay loam soil have various sites on, and we know that ethyl fluorine is highly persistent herbicide uh, because clay loam soil have various sites on which herbicides get absorbed and unavailable. Sandy loam soil and gray one is for the clay loam soil. This shows that higher amount of residues were present in recovered from the sandy loam soil as compared to clay loam soil. So ethyl fluorine is highly persistent herbicide and remain in soil for a very long time, as even after the 90 days, herbicide is not completely degraded from the soil. So it is important to remove the highly persistent herbicide by using biocomposite based strategic mitigations. Thank you.
Thank you, ma'am, for giving such a wonderful presentation. Shall we ask for another one? Okay, ma'am,
Yeah.
please remove your presentation.
Monica,
Next
can
week
you
we will-
explain,
Sure.
is it possible to do this on a large scale? Because you have done it with the 20-gram soil sample in petri dish. तो हम इसको क्या large scale पे apply कर सकते हैं?
Yes, ma'am.
तो
Yes,
अगर हम
ma'am.
large
Actually, I have done it in laboratory. I have done it in laboratory,
yeah.
and it is applied on large scale. So to get the idea कि फील्ड में ये कितना persist करेगा,
No, I just wanted to know,
उसको rate
suppose
करने के लिए.
you have a field, आपके पास एक फील्ड
Yes,
है,
ma'am.
आपने उसमें बताया ना कि हमने अपना सैंपल लिया, उसमें सिलिका जेल डाला, चारकोल डाला, मिक्स किया, एंड देन सोडियम सल्फेट डाल के उसको कॉलम से इल्यूट करके आपने अपना प्रोडक्ट निकाला.
इल्यूट कर.
है ना?
Yes, ma'am.
तो जब हम सपोज वी हैव अ फील्ड में तो बेटा हम ये
Okay.
सारा कुछ नहीं करते ना, तो अगर हमें फील्ड में इसको
Ma'am,
अप्लाई करना हुआ तो हाउ वी कैन डू दिस? आई जस्ट वांटेड टू नो दिस
ma'am.
थिंग, नथिंग एल्स.
हाँ, okay, okay, ma'am. Uh, we have potato field. Okay,
हम्म.
and हम अप्लाई करते हैं वहाँ पे इथिल हर्बिसाइड को
ठीक है.
करते हैं, वो सॉइल में जाएगा।
हम्म.
सॉइल में जाएगा तो मैम आई अप्लाइड आउटसाइड फ्रॉम ना, वो तो अप्लाई करते ही हैं, सो दैट उनका अप्लाई किया हुआ नेक्स्ट क्रॉप को कितना अफेक्ट कर रहा है, या फिर उस सॉइल में जब उन फार्मर्स ने अप्लाई किया वो कितने टाइम तक रह रहा है, इस होगा, इसका आइडिया लेने के लिए ये स्टडी है।
ओके ओके, मतलब यू जस्ट वांटेड टू से कि हाउ मच टाइम इट इज रिटेनिंग इन द सॉइल, दिस इज व्हाट दैट यू हैव डन.
Yes, ma'am. In another experiment, I have also done the strategic mitigation, but in not in this abstract I have
Okay,
submitted.
okay, no problem, no problem. Thank you so much.
Thank you.
Session is open for the questions. If anybody except ma'am want to ask any question, please. I feel ma'am nobody is asking any question. So with your permission, shall we move further?
Yeah, yeah.
Okay. Thank you, ma'am. Next speaker would be Meenakshi. Meenakshi, ma'am, are you there?
Yes, ma'am. Yes, ma'am.
Okay. Okay. Present your work.
Yes, ma'am. Ma'am, is the screen visible?
Yeah, it is visible.
Okay. Good afternoon, everyone. My name is Meenakshi, and I'm here to present my poster entitled Experimental and Theoretical Studies of a Thiophene-based chelator. So let's start with a brief introduction. Skiff-based
थोड़ा
compounds.
सा प्लीज, सॉरी टू इंटरप्ट, थोड़ा बड़ा कर दीजिए आप इसे, साइज लार्ज कर लीजिए.
Okay, ma'am.
ओके, थैंक यू बच्चा.
So let's start with the introduction. Skiff-based compounds are generally used in the field of organic and inorganic chemistry and research industry because of their capability to coordinate with the metal ions and form stable complexes. These are an important class of compounds, and their metal chelation contribute in the fields like chelation therapy, pharma, and some biological activities. So here we have synthesized a dipodal ligand that has malonamide as central unit, propylene diamine as spacers, and thiophene as the binding unit. Here is the scheme for the synthesis of the ligand. It is a two-step reaction. In the first step, diethyl malonate and propylene diamine, they react to give an intermediate. And in the second step, thiophene binding units were connected to the intermediate compound in ethanol at room temperature. So we obtained the compound with 71% yield. The compound was hygroscopic, air sensitive, and soluble in water. So various spectroscopic techniques have been used to characterize the ligand. So first of all, proton NMR spectrum. Chemical environment of the protons present in the ligand was determined through proton NMR spectra recorded in DMSO medium. Here we can see various peaks. A multiplet is seen at 1.5 ppm, which corresponds to the central methylene group of the propylene diamine. Multiplets at 3 and 3.1 ppm corresponds to methylene group adjacent to amide group and imine group, respectively. Peak at 3.5 ppm, it corresponds to the most de-shielded methylene group of the central unit because it is present between two carbonyl moieties. 3 peaks in the range of 7 to 7.5, they are present because of protons of thiophene. Peak at 8 ppm is due to C double bond N of the imine group. Means it is due to imine group. the proton is attached to the nitrogen. Now, the carbon skeleton of the ligand is determined through C13 NMR spectrum taken in deuterated DMSO. Here, peak at 166 ppm. It corresponds to carbonyl of the central unit. Another chemical shift observed at 157 ppm is due to imine group. Peaks at 59 and 37 ppm, they corresponds to 2-methylene group adjacent to imine linkage and amide linkage respectively. So another spectra is IR spectra. FTIR spectrum was obtained in form of KBr palette in transition mode in mid-IR region within the range of 4,000 to 400 centimeter inverse. Various peaks are observed. Peak at 3290 centimeter inverse corresponds to NH stretching, whereas peak at 1546 centimeter inverse corresponds to NH bending. This indicates that the intermediate molecule consists of the amide group. Another peak at 1639 centimeter inverse. It corresponds to CN stretching, which shows presence of N double bond CH group due to condensation reaction of the intermediate amine with pyrrole aldehyde. Similarly, various other peaks are also visible. Now, here we can see the potentiometric curve of the ligand and its iron complex. So the potentiometric curve obtained for the ligand shows two inflection points, one at x is equal to 1, whereas another at value 1.1. First inflection point indicates neutralization of the excess acid, and the second one indicates release of protons from the ligand. The coordination of the ligand with Fe2 is shown by deviation of FeL curve from ligand near pH 4. So ligand curve is black colored and complex curve is shown with blue curve. Now, spectrophotometric studies of the ligand and its iron complex. So in this figure, we can see the absorption spectra of the ligand that was recorded in the region between 240 to 500 nanometer within pH range of 2 to 9. Under acidic conditions, the electronic spectra of the ligand shows two absorption bands at lambda 266 nanometer and 290 nanometer, which corresponds to pi to pi star and n to pi star transitions, respectively. Here is the absorption spectra for the complex. Under acidic conditions, two bands were observed at lambda 263 nanometer and 290 nanometer. 263 for pi to pi star transition and 290 nanometer for n to pi star transitions. At pH 7, that is in neutral conditions, hyperchromic effect is observed. On further increasing the pH, absorbance decreases, that is hypochromic effect for first and second bands. Both the bands show hypochromic effect with increase in pH. Here are the species distribution curve of the ligand and its iron complex. So first we'll observe the species distribution curve of the ligand. It can be seen that at pH 2, LH4 species exist, whereas at pH 4 and 8, LH3 and LH2 species are predominantly seen. At pH 9.5, LH species is formed, whereas if we further increase the pH, hydrolysis species are observed. Similarly, if we look at species distribution curve of the complex, It shows that at pH 5, LFEH2 species exist, whereas LFE species exist at pH 6.5. Similarly, at various pH, other species exist. Now is the optimized structure of the ligand and its complex. In these structures, we can see the least energy of the ligand as well as its complex. Energy of FEL complex is much less than that of ligand, which shows that the complex is more stable as compared to the ligand. So here is the conclusion. In the study, we have synthesized and characterized a novel dipodal chelator. Ligand is light yellow in color, air sensitive in nature, and soluble in water in slightly acidic condition. Theoretical studies of the ligand and its iron complex were done by semi-empirical method using PM6 parameter. A high binding constant was obtained. Log beta value came out to be 33.39, which indicates an effective encapsulation of Fe2 by the dipodal chelator. Thank you.
Thank you, ma'am. House is open for the questions. If anybody is having any question, please ask questions. Professor Gitanjali, ma'am, you can ask any question.
No, it's okay. Meenakshi, can you tell me one thing only?
Yes, ma'am.
You have prepared the ligand with the iron. Have you tried some other metal also?
Yes, ma'am. I have also studying its interaction with other metal ions also, but log beta value came out to be maximum with iron. So it formed more stable complex with iron.
So is
Fe2.
there any reason for it? You try to find it out?
No, ma'am. Experimentally, we obtained the values and we find out that it forms more stable complex with Fe2 in comparison to other
और
ions.
metals के साथ आपने try किया है?
Yes, ma'am. We also obtained absorption spectra and potentiometric titrations with other ions also.
Okay. Thank you, Meenakshi.
Thank you so much, ma'am.
Ma'am, next. बुला लीजिए.
Next, Pratik Chatterjee.
Yes, ma'am. Good afternoon, everyone. Yeah. So
Welcome.
should I share my screen now?
Sure.
So is my screen visible?
Not yet.
Okay. So is it visible now?
Yeah. Now it's visible.
Okay.
Please
So
increase the size, sir.
sorry?
Please increase the size.
Okay.
Yes. Yeah. Okay. Good.
So good afternoon, everyone. So myself, Pratik Chatterjee. So I'm pursuing my M.Pharm from Central University of Technology and Management. It's in Bhubaneswar, Odisha. So today my topic is advanced intraperiodontal pocket drug delivery device against periodontitis. So before I start, so what is periodontitis? Periodontitis is mainly an inflammation and infections of our gum and the bone that support our teeth. So it's only due to the result is of poor hygiene and mainly it caused due to bad. Main symptom is that your bread breathe, loose teeth and bleedings, and due to swollen gums. So advanced periodontal pocket drug delivery device represent a promising innovation in the fight against periodontitis, and it is due to a severe gum disease that affects a million of people worldwide. So this is due to the cutting edge of devices provide targeted and effective treatment, and by delivering the antimicrobial agent or antibiotic or therapeutic substances, it preferably gives a compact periodontal and more efficiently, and it can be mainly done the help of a local drug delivery system management, and through like fibers or through strips or gel film system or irrigating of systems. So mainly what is periodontal pockets? So periodontal pockets are space or opening surrounding the teeth under the gum line, and these pockets can be become filled with infection causing bacteria. And periodontal pocket drug delivery system is basically a hallmark features of periodontic chronic potential, and due to the severe gum disease that mainly affects the significant portion of the global population. And these periodontal pockets are deeply due to the small gaps, which I already said, and develops between the base of the teeth and surrounding the gum tissues. These are the mainly rise and consequences of inflammatory responses and dental plaque and tartar into the chronic inflammations. And these are leads to the support of the teeth, including the gums and ligaments, and then the underlying bones. It mainly affects these parts of the teeth, and we can help through the local site drug delivery system as well, because local site specific drug delivery system will mainly help. It will quick heal the disease in the disease without giving any drug through the blood. So local site specific is like a vital component through periodontal therapy, and it will be releasing through a controlled release drug, and it will give a better result and quick result to the patient. And particularly in the management of periodontitis, it serves a gum disease characterized of inflammation and infection of the gum tissues. And these drug delivery systems are designed to deliver therapeutic agents directly to the affected gum pockets, targeting the root cause of the disease. So there are varieties of local drug delivery systems like biodegradable drug delivery systems or nanoparticles or microspheres, periodontal pocket film, and local antibiotic delivery systems. So what is my biodegradable drug delivery device? So these are mainly like small gels form or fiber or chips forms that gradually release drugs on a on a drug released over a time, that is released drug over a time. And microsphere or nanoparticle drugs, these are no, containing tiny drug containing particles, which helps in the periodontal pocket releasing drugs over an extended period. So, and the periodontal pocket films, these are mainly the thin films or strip containing drugs added to the gums and released through the medications gradually. So also we can use some local antibiotic delivery systems, antibiotic containing fibers or gel placed in the pockets to target a specific bacteria. So, and the second is, and now there's mainly the conclusion and the future perspective of this periodontal disease. So conclusion is mainly it is an periodontal pocket drug delivery device that represent a promising advancement in the treatment of periodontitis, offering mainly to the targeted and efficient delivery of the therapeutic agent. And these are mainly affect the gum pockets to show a quick results. And it also reduces the mainly uses for the treatment of reducing side effects and enhancing patient comfort, and their ability to combat underlying causes of periodontitis by addressing bacterial infection and inflammations. So the main future perspective is through personalized treatment and biological agents, remote monitoring, and minimally invasive techniques and combination therapies, and long-term efficiency research into the long-term effectiveness and safety to this device. So personalized treatment like advancement in technology may involve in customization of drug delivery to match the individual patient needs, and biological agents growth of the factors in the gene therapy in the device could open new avenues for the regenerative periodontal treatment. So in long-term efficiency, we can research into the long-term effectiveness and safety to the device, which will be crucial to the establishment to the role as standard treatment for opinion of periodontitis. So that's all from my side. Thank you, everyone.
Thank you, Dr. Pratik. It was a really interesting presentation and
Thank you so much.
today demand, because most of us are having such type of dental problems.
Yeah,
And you are
and yes,
providing a cure.
yeah. So
Nice
this is my
presentation.
actually the project, and this is a review project. Actually, I'm going to start few experiments on this as well. So these are my mainly these are the review things which I've done till now for the periodontal disease.
It's a nice topic, sir.
Thank you so much.
Best of luck
Thank
for your project.
you, thank you so much. Thank
Okay,
you.
house is open for the questions. Professor Gitanjali, if you want to ask any
No
question,
question.
please.
Actually, I just wanted to know कि आपने इसमें से कुछ किया? Have you done something? But he himself says that I have just reviewed it. So it's okay. It's good, because if you are able to at least make one of these whatever you have said biodegradable or something like that,
Yeah.
or specified any one of the device, so I think that would be much, or it will be good for the whole world.
Yeah, it is. Yeah, it is.
Yeah, because these days we are suffering a lot with this
A lot.
problems.
Sure, sir.
This is
Yeah,
a demand, na? That's why I'm saying. Yeah.
yes. Okay.
Okay, sir.
Thank you so much.
We will move to next participant,
Yes, ma'am. Yes, ma'am. Please.
Dr. Leena Priya. Are you there?
Yes, ma'am. Good evening, ma'am.
Yeah. Okay, you can start.
Okay, ma'am. Ma'am, my screen is visible.
Yeah, it is visible.
Yeah.
Good evening, everyone. My name is Leena, and I am from Birla Institute of Technology, Meshra, and I'm working on the topic sensing of nitrite ion in aqueous medium using polymetaminophenol silver nanocomposite modified graphite electrode under the guidance of Dr. Pradeep Karsar. And this study shows the fabrication of a highly sensitive chemical sensor capable of efficiently detecting nitrate and nitrite in aqueous phase using modified graphite electrode, and to achieve the uniform and stable layer of polymetaminophenol silver nanocomposite was applied onto the active surface of the graphite electrode, and using the IV technique we perform the sensing, and this technique is considered as simple and efficient method for detecting hazardous chemicals and environmental pollutants among various detection techniques. So this is the synthesis procedure. In First, we synthesize the silver nanocomposite containing 5 weight percent of silver to obtain core shell morphology, and then we characterize the same using different analytical techniques like ICP, OES, FTIR, FESAME, EDX mapping, TAME, XRD, source meter for DC conductivity, and then we proceeded for sensing of different analytes of known concentration.
This is the first scheme, and this is the mechanism of polymetaminophenol silver core shell hybrid formation. This mechanism of the silver nanocomposite in an aqueous NaOH medium, and the metaminophenol phenoxide ion formed complex with the Ag plus of silver nitrate in DMSO water mixture. And the oligomeric shell of metaminophenol was formed through oxidative polymerization by the reduction of Ag plus to Ag0 core upon refluxing at 80 degrees Celsius. Whereas this is the table 1, which shows the which confirms the loading of silver nanoparticles, and it was around 6%. In figure 1, this is the FTIR spectra of polymetaminophenol silver core cell hybrid. From FTIR spectra, there is a free NH groups of polymetaminophenol, and without having any intermolecular hydrogen bonding in polymetaminophenol silver core shell hybrid, it appeared at 3230 centimeter inverse, and there was two strong bonds which appeared at 1563 and 1493 centimeter inverse, and due to stretching of O and N of O quinonimine units, and it is clear that O quinamine units were formed in polymetaminophenol core shell hybrid during the reduction of Ag plus to Ag0. Whereas in the XRD pattern of the polymetaminophenol core shell hybrid containing silver nanoparticles, the formation of Ag plus nanocrystals was confirmed from the characteristic XRD pattern of the nanocomposite, and the characteristic peaks value obtained at 2 theta values, which are 38.1, 44.1, and 64.5, including 77.6. And the diffraction planes are shown in figure 4. Through DLS, we confirmed the size of the silver nanocomposite, which was around 91 nanometer, but I haven't shown here due to lack of space. And from the FE-SAME and TAME image, we confirmed the morphology and the exact size of the particle. There's the thin layer of the polymetaminophenol coating wrap, or we can say wrapping on the outermost layer of silver hybrid. And to have a polymetaminophenol silver core shell structure of synthesized hybrid, there is a small spherical beads which is shown in figure 2, left hand side. It's a core shell structure with some bulk polymer coating were confirmed in the HR-TAME image. From that, we calculated the thickness of the shell. It was around 220 nanometer. This cell surrounds a uniformly distributed silver core with a size ranging from 35 to 40 nanometer. It should be also around 30, we can say that it's 30% bulk polymetaminophenol, and which was distributed around the core shell structure. And the right hand side picture shows the EDX mapping, which clearly reveals that the distribution of the silver nanoparticles within the polymer matrix of the polymetaminophenol silver core shell hybrid. And this is the conductivity of the silver nanocomposite, which was around 10 to the power minus 7 Simon centimeter inverse. Now I'll be coming to the sensing of nitrate and nitrite salts. So in figure 6, this is the sensor setup in which the modified graphite electrode was used as working electrode, and gold electrode was used as counter electrode, and both the electrode was connected to a source meter and was immersed in the analyte, and increase in the current was observed by applying the voltage, And to prepare the sensing analyte, the required concentration of salts of the respective ions like ammonia, nitrate, nitrite, sulfate, chloride, carbonate, and urea was dissolved in 10 ml of water at room temperature. Whereas the figure 7 and 8, whereas the figure 7 shows the typical electrical response of the IV curve of the nitrate chemical sensor fabricated using silver nano composite, and the response demonstrates both in the absence and the presence of a nitrate and nitrite in water. And upon introducing the target analyte, especially nitrate and nitrite ion, resulted a noticeable increase in the current. And this enhanced current indicates the sensitivity of the polymetaminophenols silver core shell hybrid modified graphite electrode towards the analyte. And the figure 8 shows the calibration plot where good linear relationship was obtained for sensing of varying concentration of nitrite and nitrate at 4 volt. And this is the cross selectivity which was observed for successful sensing of nitrate in presence of the other anion. And the mechanism was confirmed by the zeta potential values. According to the table 2, for different analyte, and there are different values for different analyte with the silver nanoparticle. And the change is attributed to the interaction of the polymer with the water. And in water, the polymer and the nanoparticles do not separate due to the strong association between them. And upon adding the nitrate ions such as sodium nitrate, ammonium nitrate, potassium nitrate to water, the zeta potential values ranges from -10 to +10. This is due to the formation of strong hydrogen bonds. And this is the sensing mechanism of the nitrate, where we confirm this is due to the additionally negative charge of the oxygen atoms in the nitrate ions, stabilizes the silver nanoparticles, whereas the sodium carbonates and ammonium hydroxide, being basic in nature, exhibit negative values due to the presence of very weak hydrogen bonding. And ammonium chloride, being slightly acidic in nature, results in increased value due to the presence of weak hydrogen bonding. And very good response was observed for nitrate and nitrite. Thank you, ma'am.
Thank you, ma'am, for your interesting presentation.
Yeah, I just wanted to know one thing.
Yes, ma'am.
Up to which
Sure.
micro concentration or molar concentration you are able to detect the presence of nitrate or nitrite?
Ma'am, we used 5, ma'am, the results was very good in case of 5 millimolar.
5 millimolar.
Yes, ma'am.
So if some other ion like phosphate, etc. is present, it gives the same result?
No, ma'am, the current is very less in that case.
Okay. And without this electrodes, it is not possible to sense.
No, ma'am, we need a modified graphite electrode. Without the sensing material, we can't perform. We also perform the sensing in case of bare graphite electrode. The result was very bad.
Okay. Good work. Thank you.
Thank you, ma'am.
Okay, ma'am. Shall we go further, ma'am?
Yes, yes, please.
Next is Manisha Huda. Are you there, ma'am?
Yes, ma'am.
Okay. Start.
Okay, ma'am. Good afternoon, everyone. I am Manisha from Baba Masnath University, Rohtak, Haryana. Today I am going to Ma'am, my screen is visible.
No, it's not there.
Is it visible, ma'am?
Yeah.
Okay. So I'm going to present my poster entitled Synthesis and Biological Activity of Azole-based Heterocyclic Derivatives. The rise of multi-drug resistant pathogens present a looming threat in the medical world. Despite our scientific progress, infectious diseases remain a significant challenge. Understanding this, our primary motivation was to identify and synthesize novel antimicrobial agents which could offer a fighting chance against these resilient pathogens. So, our objectives are to delve into the world of azole based antimicrobial heterocyclic derivatives with a focus on reviewing recent advancement in this field, synthesizing imidazole-based chalcone agents, creating innovative agents integrated with isoxazole and pyrazole on an imidazole backbone, characterizing and conducting antimicrobial studies of these agents. Now, the methodology includes 5 chapters in which the chapter 1 deals with the advancement in thiazole-based antimicrobial derivatives. In this, we are going to review regarding thiazole-based antimicrobial derivatives. In next chapter, we are going to synthesize imidazole-based chalcone derivatives. With our foundational knowledge established, we will synthesize our material compound 1, which is shown in the scheme, and then we will synthesize our compound 2, which is imidazole chalcone. And in the third chapter, we will introduce isoxazole to imidazole derivatives. In this, with the base hydroxylamine, we are going to introduce isoxazole in moiety to the imidazole derivatives. In next chapter, we are going to introduce pyrazole to chalcone derivatives outlined in this scheme. And next chapter, we will deal with antimicrobial studies of thiazole, isoxazole, and pyrazole agents. Now, the outcomes were synthesis promise regarding antimicrobial testing. We anticipated that our derivatives will display notable antimicrobial activity against common pathogens. And if I conclude, the synthesis and evaluation derivatives are critical for future antimicrobial drugs, and preliminary findings indicate promising directions for continued research and potential clinical application. So basically, we are going to synthesize azole-based heterocyclic derivatives, which will work against the pathogens. This is all about my topic.
Okay, thank you, ma'am.
Thank you.
So you will synthesize all these compounds. Have you synthesized any one of them?
Ma'am, this is on the basis of my review work. I'm going to synthesize these.
Okay, you haven't synthesized any compound up till now.
No, ma'am.
Okay. So this is actually your chapters for the PhD.
Yes, ma'am. I'm
Oh.
going to work on it. Yes.
Okay.
This might be your first year, right?
Yes.
Best of luck.
Thank you, ma'am.
Ma'am, next बुला लीजिए.
Yeah. Next is Leslie Lovely. Are you there?
Yes, ma'am.
Okay.
Okay,
Please
so
start.
good evening, ma'am.
Good evening.
Is my slide visible?
Yeah. It is.
Okay, ma'am. So my name is Lacey Loveleen, and currently I'm pursuing my PhD under the guidance of Dr. Surendra Nimesh at Central University of Rajasthan. So, ma'am, we are dealing with the antimicrobial properties of biosynthesized silver nanoparticles. And sorry, uh, sorry, ma'am. Okay. Okay, so we are dealing with, actually we are synthesizing the silver nanoparticles that are being derived from Terminalia bellerica leaf and bark extract, and we are using those silver nanoparticles to test over the bacterial cells of 4 different kinds of bacteria, as in they are removing the resistance of those bacteria or not. But the study is now in silico form. Further, we are going to deal with in vitro and in vivo analysis as well. So the concept is that suppose here is a bacterial cell and it has many components like cell wall and cell membrane, some transport channel proteins, efflux pumps, and some other integral proteins. And what happens that once a drug enters into the bacterial cell, it starts to do some alterations inside the bacterial cell. But in the case of multi-drug resistance, what will happen that one of these channel may alter and the inflow of drug inside the cell stops, or the efflux pump number increases, through which the efflux of the drug increases from the bacterial cell. So what our nanoparticle does is, the nanoparticle is basically a depot of many of the silver ions inside a spherical shaped nanoparticle. And in the case of green synthesis method, many of the phytochemicals that are present in the leaf or the bark extract of Terminalia bellerica would cap over that silver nanoparticle. So those phytochemicals will act first over the outer surface of the bacterial cell. Further, it will disintegrate and the silver ions would inhibit the functioning of bacterial cells. So this is the whole idea. And based on this, we have some of our objectives. That is, we would first go for in silico analysis, and for that, we are using Autodoc version 4.2 tool for docking purpose. So in this tool, first of all, we need to prepare our target. Our targets are the bacterial proteins that are present over the bacterial surface or the membrane proteins of the bacteria. After that, the ligands. The ligands here are the phytochemicals that are present in the Terminalia bellerica extract. So these preparations are done in a 3D model. After that, the binding sites are determined. So what is basically a binding site? Here you can see that in our target protein, there are some of the amino acid that forms a binding site where a ligand binds. So we need to determine our position where probably our ligand could bind. So that is determined. Next, the grid is prepared. Grid is a 3D structure that binds or bounds the binding sites. Indirectly, we can say that it forms a coordinate where our ligand could bind in the given protein. Then we are going to determine the interacting positions or the interacting amino acids. After that, we are going to see the binding energies and then the docking structures. So here is a model that how a phytochemical interacts with bacterial protein in different runs. So further in this study, based on this study, we have some of the conclusions. We have found through literature some 8 proteins from 4 strains of bacteria, that is Escherichia coli, Pseudomonas aeruginosa, Staphylococcus aureus, and Bacillus subtilis. Their binding sites were analyzed. Further, their binding energies were also studied. This one is for the phytochemicals that we had chosen, and this one is for the silver nanoparticles. So what we found that in case of phytochemicals, this is a pictorial representation of the interaction. We found that in the extract, in the whole extract, from various different phytochemicals that were present, a particular phytochemical, that is this argingenin, it interacted with almost 4 out of 8 proteins that we had selected earlier, having a very high binding energy. followed by the interaction of silver ions with different proteins. Here we have taken a single atom of silver. Reason is that once the nanoparticle interacts, it disintegrates and releases the silver ion. So for that purpose, a single silver ion was chosen for the interaction, though the ion is very small, that particularly it is not getting entrapped into any of the protein. And secondly, through in silico analysis, the autodope doesn't show any metal complex interaction. It is not available in its software. It only shows for hydrogen bonding. So this was all about my presentation. And we have done the in silico studies till now, and further we have characterized our silver nanoparticles, but the data is yet to be completed. Thank you, ma'am.
Okay, thanks, ma'am. Nice presentation.
Thank you, ma'am.
So anybody want to ask any question?
Leslie, have you done any in vivo studies?
Ma'am, not in vivo, though we are trying to do it in mouse model very soon, but we have performed some of
I
the
just wanted to know, you will do all these studies in just by docking, or you will be performing any practicals in the lab also?
No, ma'am. We have performed some of the practicals, like we have performed the disk diffusion assay and MIC-MB calculation, but actually the data is not, yet to be completed. So that's why I have not shown here the incomplete data.
You have used the plant Terminalia balearica.
Yes, ma'am.
So have you heard about Terminalia arjuna?
Yes, ma'am. Yes, ma'am.
So can you tell me what is the difference between these two?
Ma'am, actually, if we go through the phytochemical analysis, then by phytochemically, they are quite similar. So, through previous literature, we had seen that when antimicrobial property was studied for both the plant extracts earlier, it has been found that not for nanoparticles entrapped, but only the extract, it was found that balearica had a more potent antimicrobial property compared to Arjuna. So that's why this plant was further chosen.
Okay, thank you so much.
Okay, ma'am, thank you.
Thank you, ma'am. And this is the end. Like we were having 8
Two more.
candidates and 2 were absent.
2 are absent, no?
Yeah.
1, 2, 3, 4, 5, 6. So I just need to send you the results, or
Yeah. You have to send the results.
okay, then I will just compile and send you.
Not right now. First, I will give you a vote of thanks, and then they will leave. Then you can send.
No, I just wanted to know, here only we need to announce, or I will just send you and then you can send them.
Ma'am, just send it to me.
Okay, no problem.
They will get the mail.
Okay. Thank you so much.
Okay. So I stand before you today for giving the vote of thanks with immense gratitude and a heart full of appreciation as we come to the close of this remarkable second day international conference event. It has been an honor to serve as your host and deliver this vote of thanks on behalf of our entire international conference college community. First and foremost, I would like to extend my heartfelt thanks to our distinguished chairperson and our speakers for gracing this occasion with their presence and wisdom. Your guidance and support have been invaluable in making this event a resounding success. I would also like to give a special thanks again to our chairperson who has given lots of time to us from our precious hours, and all those who contributed their talent and expertise to make this event both enlightening and entertaining. Your contribution have added depth and color to our program, and we are grateful for your participation. Thank you all.
Thank you, ma'am, for such a nice host. Thank you so much.
Thank you.
And
You
all
are
the best
wonderful,
to all
ma'am.
the participants.
Thank you, ma'am.
So you all can leave, and result will be announced on your mail. You will get the mail from our side, and by tomorrow, you means out of all you, 3 are being selected, and you will get the information on your mail. Now you can leave. Thank you.
Thank you. Okay.
Professor Gitanjali ma'am, you are not supposed to leave.
Okay, no problem.
So ma'am, you there in English Bandhu, right? Please leave. उनको leave कर कराओ. खुद भी leave करो. आता है?
Shall I leave, Anjali?
हेलो मैम.
हाँ जी मैम.
हाँ जी मैम. मैम कौन सा सबसे अच्छा लगा आपको?
देखिए मेरे हिसाब से इसमें अगर आपने देखा होगा
हाँ जी, हाँ जी.
तो ये 3 बच्चे थे जिनका लैब वर्क था. एक मोनिका का, मीनाक्षी जिसने बोला था मेरा नाम हटा दीजिएगा, नैन.
हाँ हाँ हाँ.
और ये जो तीसरी बच्ची थी लीना प्रिया.
ओके.
इन तीनों
लीना
का लैब
प्रिया.
वर्क. हाँ जी.
ओके ओके
तो लीना और मीनाक्षी वर बेटर.
ओके.
दोनों ही ठीक थे. बिकॉज
ठीक
लीना
है.
का काम भी अच्छा था और मीनाक्षी
अच्छा
का
था.
काम भी अच्छा था, और दोनों का मतलब प्रेजेंटेशन वाज आल्सो गुड. लीना का a little bit better था in comparison to मीनाक्षी. अदरवाइज both have worked in the lab. तो आप
ठीक
देख
है.
लीजिए.
नहीं
और
नहीं,
दो
ठीक है
मेरा
मैम. लीना लीना मुझे भी ज्यादा अच्छा लगा इनका ना, क्योंकि इन्होंने काफी
हाँ.
इलेबोरेटेड वे में बताया भी काफी अच्छे से था, है ना.
हम्म.
और एक्सपेरिमेंटल वर्क काफी अच्छे से
हाँ.
एक्सप्लेन किया था. ठीक है,
बाकी
तो
बच्चों का
लीना
तो रिव्यू ही
फर्स्ट।
हो।
हाँ
और
जी,
सेकंड आप रख लीजिए मीनाक्षी को,
ठीक है, और थर्ड
फिर मोनिका को रख लीजिए, बिकॉज बाकी
मोनिका।
बच्चों का तो रिव्यू वर्क किया है। हाँ जी,
ठीक है, ठीक है,
आप
मोनिका
बताइए।
थर्ड।
हाँ जी,
हाँ,
बिकॉज बाकी बच्चों ने तो रिव्यू किया है, जैसे प्रतीक का भी रिव्यू है।
रिव्यू ही था। हाँ, अगर
हाँ जी,
प्रतीक ने कुछ किया होता ना, जैसे वो
हाँ
उनकी
उसका
अभी
काम
है,
अच्छा था।
काम अच्छा है, थॉट अच्छी है, लेकिन अभी किया नहीं है, तो अभी ग्राउंड पे कुछ है नहीं। ठीक है, मोनिका का ठीक है मैम।
ठीक है
इनका
ना।
भी लवलीन का भी काम मतलब उतना वो नहीं था, प्रेजेंटेशन ऑल्दो अच्छा था।
हाँ प्रेजेंटेशन इज गुड, बट उसने भी काम लैब में तो
हाँ,
नहीं किया।
नहीं नहीं किया वो। ठीक है मैम, ठीक है। तो
ठीक है ना।
लीना फर्स्ट एंड सेकंड इज मनीषा एंड थर्ड इज मीनाक्षी। ओके मैम।
नहीं नहीं नहीं, लीना, मीनाक्षी एंड मोनिका।
लीना।
लीना, लीना,
हम्म।
मीनाक्षी
एंड मोनिका।
एंड मोनिका।
ठीक है, ठीक है, ओके।
ठीक है ना, ये मुझे
ठीक है,
आपको लिख कर कहीं अलग से देना है।
नहीं 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 नहीं,
ठीक है ना।
मैं हाँ हाँ, मैं पहुंचा दूंगी। मैम
ओके
आप
ओके, थैंक।
देशबंधु में थे पहले, आपका मैं पढ़ रही थी।
हाँ
यू
जी,
वर
मैं
देयर इन देशबंधु।
2004 टू नवंबर 2004 टू मार्च 2006,
ओ,
ऑलमोस्ट वन।
ऑलमोस्ट 1 ईयर।
हाँ
ओके, मैं आपके देशबंधु में 2010 से हूँ मैं।
ओके। नाइस टू मीट यू, इसी
सेम
बहाने ही
हियर।
सही।
बिल्कुल, लेकिन आपका डिपार्टमेंट केमिस्ट्री एंड माइन इज फिजिक्स।
हाँ, बट वहां पे तो केमिस्ट्री फिजिक्स ऑलमोस्ट
सेम।
सेम, नीचे ग्राउंड फ्लोर पे ही है
हाँ,
ना,
हाँ जी, हाँ
एडजेसेंट
जी, हाँ
ईच
जी, हाँ
अदर।
जी। हाँ, सिस्टर्स एंड सिस्टर्स हो गया। ओके,
चलिए, यू पीपल आर
नाइस टू
डूइंग
मीट यू।
हार्ड वर्क।
अरे मैम, बहुत अच्छा लगा मैम आपसे मिलके। आपका प्रोफाइल में पढ़ रही थी, आपने तो बहुत काम किया हुआ है, बहुत बहुत बहुत।
नहीं नहीं, ऐसा कुछ नहीं है, इट्स जस्ट अ स्मॉल एफर्ट टू डू समथिंग।
ना 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 ना, काफी अच्छा है मैम। हम लोगों का भी क्रॉलिंग स्टार्ट हुई है। आई डिड माई
नहीं, वो
पीएचडी फ्रॉम
स्लोली है।
जापान। ना ना,
ओके,
अभी स्टार्ट हुआ है। अब देखते हैं कहाँ तक जाता है।
तो जाएगा अभी।
थैंक यू फॉर योर ब्लेसिंग्स। देखते हैं क्या होता है। कभी
अच्छा ही
कॉलेज
होगा।
आएंगे ना मैम, देशबंधु आने का कभी भी टाइम मिले तो प्लीज एक बार कॉल कीजिएगा, तो मैं आपसे मिलने आऊँ। या अगर
ओके,
मैं किरोड़ी में लाती हूँ तो तब मिलने आऊँ।
हाँ पक्का।
ठीक है।
ओके
चलिए,
ओके
ओके
ओके,
मैम,
थैंक यू सो
थैंक यू,
मच।
थैंक यू।
ओके
थैंक यू।
ओके, बाय।
ओके, बाय।